LTR is also relatively young. And so you can see these clusters. The point is, with these arrows, is to indicate that these clusters are brought about by different waves of transposable elements in our genome. And multiple waves just incorporate into our genome. And if you look at them collectively, group 3 sites, both CCREs and transcription factor binding sites, these are the smaller sites. And group 3, like these are the blue ones, they are brought about mainly by transposable elements, these youngest families. The line 1, LU, LTR, these are the youngest families in our genome, and they are the ones that have been spreading these group 3 sites across our genome. And that's why you see them as individual clusters, is because they are brought in by TEs. And one interesting thing about these kind of display for transcription factors is not only group 3 sites are discrete clusters, even other groups, not group 3, they start to show lobes of structure. Here is FOXA1, which is a pioneering transcription factor, and this is like 700,000 FOXA1 sites. And we were very intrigued by these individual lobes. Lobe 1 here are the ones that are across, red is present, blue is absent. Now this is the entire tree of 240 mammals, and they are present in all but bats. They're gone in bats. Those are the cluster. And then 2 is in New World monkeys, and different lobes indicate different losses in the evolutionary history. And one thing that was very interesting is lobe 4 here are sites that are gone in even-toed ungulates and cetaceans. So cetaceans are whales and dolphins, and even-toed ungulates are like the cows and those domestic animals that have even number of toes. And they are related in the evolutionary tree, and this is really fascinating to me because cetaceans are really like mammals that are sick and tired of land life and then went back into the ocean, and they show up as a cluster of 4 here, and there are some transcription factor binding sites that are lost in this lineage. So we had lots of fun about transposable elements spreading these sites. But as I motivated at the beginning of my talk, we were truly really interested in annotating the functional sites that might explain the relative traits and the relative diseases. Right? So we were always wondering, which end of our sites could explain the difference between human individuals in terms of susceptibility to traits. So Greg devised this technique to separate the transcription factor binding sites into 2 portions. One is constrained, one is unconstrained among the 240 mammals. And the reason he did this is because you have to do this for individual transcription factors. Their evolutionary speed is quite different between different transcription factors. So there are 2 examples here, like YY2 and HNF4A. So you see for YY2, the portion of the sites that are constrained or conserved, for whatever purpose of our discussion, constrained is the same as conserved. So these conserved sites across mammals is a bigger portion for YY2 than for HNF4A. You see this bump here is a lot smaller compared with the neutrally evolving portion. And the neutrally evolving portion, you can see that it has a vital P score around 0. And then the conserved ones have much higher vital P. And like this is average vital P for the conserved ones is much higher, and then it peaks at the binding site and correlates with the high information content of these positions in the site. So we had to do this for individual transcription factors. So we have these two sets of sites, right? So we're trying to find out what's the difference between them. So there are a lot of detailed analysis that I'm not going to drag you into, but the point I want to make is the constrained sites are the sites actually that are really what matters in a lot of the things I'm going to talk about. Constrained sites are like the tip of this group 1 here. The reason that the group 1 site, the difference between the group 1 site and the constrained site is, for the constrained site, we actually take into account the speed of mutation rate by using phylo P. This is just number of species that are conserved. It doesn't take into account the speed of mutation. So that's why these ones are a better measure. So a number of analyses we did to show that these constrained sites are more likely to be functional in individual species. So we looked at a bunch of species here with ChIP-seq data in the individual species, and the sites that are conserved, constrained sites here have higher logos, like unconstrained sites. When you go to more distant species, the logo becomes shorter and they're likely to be bound. And we looked at the ChIP-seq data sets of the same transcription factor in each of the species, and the sites on the top are the constrained sites, and they're more likely to be bound in the respective species, and the sites that are non-constrained are less likely to be bound. And then we can also look at the footprints of DNAs. So usually when you have a transcription factor that's found in a location, it protects the DNA from being cleaved by the endonuclease DNase 1. So you see this footprint. And the constrained sites are more likely to leave footprints in the respective cell type. So basically we did a bunch of analysis to show that these sites are truly more likely to be functional. So let me just bring home in like 5 minutes of my talk to what I started out. It's a very long journey. We were interested in GWAS variants, right? The genetic variants that might explain human trait and the phenotypes. So how can we use our annotations of these regulatory elements and the smaller TF binding sites? These are basically components of CCREs. How can we use these two sets of regulatory elements and sites to annotate the genetic variants that are associated with traits and diseases? So we for ENCODE 3 paper, we systematically looked through the catalogs by these NHGI, EBI, GWAS catalog. At that time, it had 3,800 studies, and now it's more like 10,000 studies if you go to the same catalog. And we can look at the variants, and then we have to take into account this linkage, this equilibrium structure in the human genome. Once that's taken into account in LV, I'm not going to explain this, I don't have time, but you need to do that. And after you have taken into account that, we can basically try to identify which biosamples are more likely to be relevant for a particular disease, which individual variants are more likely to be functional. So here is like an example that we wrote up in the Nature paper about how we can use CCREs to annotate individual SNPs, single nucleotide polymorphism. So here is an example of a SNP that's associated with schizophrenia. You can basically, the SNP is here, and you can try to figure out. I started by saying if the SNP does not overlap any coding gene, there's very little you can do unless you delve into regulatory elements. So here is a SNP that does not overlap coding genes, coding exons, I should be more precise, and it doesn't even overlap a CCRE. However, it's in high linkage disequilibrium with other SNPs which overlap CCRE. So you have to jump a few steps And you take the CCRE data, which we have, across all kinds of cell types, and start to make hypotheses about which ones could be functional. And in this case, we followed one CCRE across human and mouse functional data through developmental time points, and even in collaboration with Len Panakios' lab to perform transgenic mouse experiments to show that a particular CCRE that's connected with this schizophrenia SNP might be bound by the SP3 transcription factor, and the variants associated with schizophrenia might impact binding of this transcription factor and might have a function in the brain. So, so I'm going to skip this because Henry can talk about screen. So at the end, I just want to bring home all the things I've been telling you, hopefully not so disparate. So as part of the Zoonomia consortium, we define these transcription factors that are constrained, right? So we want to see whether or not they can explain human traits. So there is this analysis you can do is linkage, LD, the same linkage disequilibrium, LD score regression across diverse human traits. So we used a collection of 69 diverse traits that are very well curated by the LD score regression people, and basically this is a bar plot to indicate how much the regulatory regions in the genome can explain the heritability of these traits as a collection. So here are the ENCODE CCREs. Here is like where we start. Like coding is here. Okay, so you just need to focus on how tall the bar is. So coding, transcription start site, EQTLs, these are the ENCODE CCREs. And these are the Zoonomia constrained nucleotides. They do better than the previous annotations. Here are basically the two main features in the Zoonomia main papers. And we have a companion paper that's going to hopefully come out soon, early next year. So the point is the highly conserved transcription factor binding sites I told you about, the constrained ones. So those are the ones that explain more heritability of these diverse traits. And they go above and beyond the positions of the Zoonomia nucleotides. We can slice and dice our transcription factors by conservation. And if you go down in conservation percentile, you can see that most of the signals come from the conserved portions. And without going into detail, here is a plot that shows the cell type specificity of the traits. And for the immune traits, immune cells transcription factor binding site has the most enrichment. And for red blood cell traits, this is K562 is the erythroid cell line that explains the most. So, okay, so there's a very long summary as to what we do. I'm not going to go through it because of the time interest, but I just want to, for every talk, I hope you have some take-home message. There's a very short take-home message. Okay, so if you look at the human genome, we have annotated regulatory elements in two scales. The bigger ones are the CCREs, bigger in terms of 200 base pair-ish elements. And then also the smaller ones, it's transcription factor binding sites. We have annotated them across many ENCODE cell types. So you know, we obviously try to impress you about how much work we have done. But the whole point is, if you look at them collectively, roughly 1/5 of these elements are transposal innovations in the primate lineage. So we've been very busy inserting new things into our genome by transposable elements. Okay. However, no matter how fun those things are, If you care about explaining the human traits and the diseases, you really need to look at the other extreme of the spectrum. The most deeply conserved elements across mammals, which are roughly 1/8 of all of the elements I've been telling you about. These conserved ones are the ones that are most likely to regulate the diverse human traits. Okay, so that's basically it. And I think I mentioned to everybody who did the work. Obviously, it's not possible for us to do what we do without the beautifully made data by the ENCODE consortium. And we're also part of Astronomia, and then Henry's going to go into these tools